So in this particular chapter, we're going to look at how we get to the diversity of life forms that we see today, how life was thought to originate, and why we see so much diversity, like we see in this coral reef here, and like we see in the fossil record of things that were once existed and are no longer alive today or have gone extinct. So your learning objectives for the first section, which is titled Deep Time, we're going to look at describing the Earth's history, be able to do some of the basic understanding of what are some major events that occurred there, and then understand the relationship between these geological events and the evolution of life. And the diagram that you see here does, the original one does show life forms as we see them appear in the fossil record. I removed them now for the discussion of just geological time. So in the diagram to the right, it's kind of small, but we see that that geological time is very, it's immense. You got to go back not 1,000 years, not a million years, but billions of years, and a billion is 1,000 million, so it's a long period of time. And we can break that up into 4 eons. There's eons, there's eras, so you can break up your eons into eras, and then you can break up the eras into periods, which we see in the third column here. The important thing I need to point out here is that there's a scale change right here, and if you go back this way here, that represents about 4 billion years of time. So we go all the way back to here, that would be 4 billion years ago, and then we come to about right here, you are like 500 million years ago. So going forward this way all the way to present, that only represents about 0.5 billion years of time, which is 500 million years, right? So the point here is that from here going back in time this way, that amount of time is actually 8 times greater than what you see above the line. So in other words, the diagram changes scale on us. Something you really shouldn't do when you're graphing unless you really highlight that to the reader of your graph, because visually it looks deceiving. Every one of these major marks here represents 500 million years of time, 500 million years of time. In other words, this one amount of time we see above the line, that's what each one of these represents. Okay. Then above here, we go to a smaller unit, and we're going only every 50 million years here. So keep that in mind, right? So again, we need to be able to understand that Earth's history dates back 4.6 billion years based on repeatedly measurable evidence. If we go to the first 500 to 700 million years, first 500 million years or so, we're going to call that the Hadean eon. Hadean, like Hades, that mythological place that's similar to what people might refer to as hell. And here, during this time, no life probably was possible. There's no record of any rocks from that period of time because of events that occurred then, and also because of the rock cycle we see today. But the Earth is getting smashed with asteroids, and it's thought that one large asteroid did hit the Earth with enough force to not just create so much energy that the Earth heated up to 200,000 degrees Celsius, but also create enough debris to leave the Earth to actually form what we see as the Moon today. So that is certainly a very interesting hypothesis with some evidence to support it. So here's what it looks like with the life forms as they appear in the fossil records. And so I want to keep this in mind here, that anything before this here represents that 4 billion years of time. And our 4 eons are, let's zoom in here. And I'll tell you what I'd like you to know for now. So the Hadean is the first 500 million years. And you have the Archean. One thing I want you to know is this is when we find the first life forms. So I'm going to go back and make a little bit of notes on here because I can't write on this right now. And then there's the Archean. And then we have the Proterozoic, which literally translates to first life. And there you see the first eukaryotes. Eukaryotes, you know the difference from your biology 1 course. Prokaryotes are like the bacteria. Eukaryotes are more complex. Cells about 10 times bigger than a prokaryote cell. They have a nucleus and so on. You're also going to see the first multicellular organisms evolving in here. And then as you come here, you're going to have You go into the Phanerozoic eon, and that's broken up into eras: the Paleozoic, ancient life, Mesozoic, middle life here. And there's where you have the familiar dinosaurs. The Mesozoic, or probably the Jurassic Park you heard of, Triassic, Cretaceous. The end of the Cretaceous is when the dinosaurs go extinct, and then Cenozoic, or modern era, or modern time here. So some things that I think you should know. First of all, is that the first life forms are seen in the fossil record approximately 3.5 billion years ago. So first life, evidence of first life, 3.5. Earth's formation, 4.6 billion years ago. Don't get those two confused on the question. Pay attention if I'm asking about the age of the Earth or when the first life forms occurred. The other one I want you to know right here is the Cambrian. Because of the Cambrian explosion here, you have multicellular organisms, but around this time during the Cambrian period, you start to see lots of diversity of fossil multicellular organisms. All animals that we see today can be traced back to the evolution of animal life, for example, which are multicellular during this time. And that is about 500 million years ago, or half a billion years ago. And that is at the start of the Phanerozoic eon. So when we look here at, so know those, and they're going to come up again here and there. So during this time, an important player in changes that occur on the planet is carbon dioxide. And carbon dioxide is important because carbon dioxide is a greenhouse gas. And that means that it can absorb heat energy called infrared from the sun. As it comes in, it absorbs it as a molecule, causes the bonds to bend. Just like a spring, you bend it, it requires energy to bend it, not break it, but bend it. And then if you let the spring go, it's going to release mechanical energy. Same thing when the molecule goes back to its normal shape, it re-radiates the infrared, which then gets picked up by another CO2 molecule. So the end result is carbon dioxide is bouncing back and forth between the molecules, and that traps heat and keeps Earth from becoming a snowball, completely frozen over. Okay, so the atmospheric levels of CO2 have changed, and the origin of carbon dioxide probably from volcanic activity and from the hot heated rock itself. Water is probably also present in these rocks and gets released. So during times when temperatures are warmer, so we're talking about cooling down after the Hadean, where it's possible to have liquid water form. And so how much liquid water you have depends on how much CO2 you have in the atmosphere. And so more CO2, carbon dioxide, is going to cause higher global temperatures. So we've probably heard of global warming before. And because of the greenhouse nature of the gas. And so if you have lower CO2 levels, then Earth becomes cooler. In fact, it can become so cool that you get massive sheets of ice on the surface of the planet called glaciers or glaciation. And even in some instances, completely snowed over or frozen over, called snowball Earth, right? Now, here's the thing is, when temperatures are warm enough to allow for liquid water to form on the surface, then that water can vaporize as it absorbs radiant energy from the sun, and then it becomes the clouds up on top. Now, what happens here is that the carbon dioxide in the atmosphere, it's a gas in the atmosphere, reacts with water vapor that's also in the atmosphere, or the little droplets up in the clouds there, and that's going to form carbonic acid, H2CO3. Okay, that carbonic acid, like any other acid, is going to break apart to form protons or hydrogen ions, and your bicarbonate ion, HCO3 only. HCO3 with a negative charge. So this is your bicarbonate ion, which is mentioned right here. Now that bicarbonate ion, and keep in mind that the carbon dioxide gives us carbonic acid. So the carbon that's in there comes from the carbon dioxide. So there's no longer do you have carbon dioxide, it's now mixed with water and reacted with water in a chemical reaction to form this. So what is that doing? That's pulling carbon dioxide out of the atmosphere, which means you would have less carbon dioxide trapping heat energy. Now, the bicarbonate, which is now in the water, it's aqueous, it's mixed within the water. That water vapor condenses and forms rain. When it comes down and hits the Earth, It's slightly acidic, but even if it wasn't slightly acidic, water, when it hits rock, can cause erosion. Mechanical erosion of the rock, cause the rock to dissolve as well. Just physical changes. But another thing that happens is if it's slightly acidic because of the weak carbonic acid, that can cause minerals to dissolve that release calcium ions. This is written incorrectly. It's not a subscript. It should be a superscript there. And calcium has a +2 charge on it, and it can react with that bicarbonate ion and form a solid. So these are aqueous. They're dissolved in the water. And it reacts to form calcium carbonate. And calcium carbonate is found in things like caliche, limestone, marble, and so on. And what happens is that calcium carbonate solidifies, it precipitates, falls down to the bottom of the ocean, and then as the sediments start packing on top of it, forms things like limestone. But here's the point. There's the carbon dioxide. The carbon dioxide is part of this substance now. And what that does is that ends up removing carbon dioxide from the atmosphere. And what does that do? That ends up lowering atmospheric temperature. So temperature would decrease because of this process. So the weathering of the rock can create lower temperatures. And so we're going to have these cycles of loss of CO2 from the atmosphere and getting colder, and then more CO2 into the atmosphere, maybe from volcanic activity, and then warmer temperatures over long periods of time, not over 250 million years of current climate changing that we see occurring after the Industrial Revolution. There's a big difference in this occurring over deep time and short periods of time in the idea of global warming and climate change we see today. So this process is highlighted here in the diagram. You can see CO2 up there in the atmosphere mixing with the water in the clouds. Up here at top, forming your carbonic acid, and then as precipitation occurs, the water, rain, we get that weathering of the rock, and there you're going to have that dissolved bicarbonate ion and the calcium cations there reacting to form your solid calcium carbonate, which precipitates to form solid sedimentary rocks. So another thing to keep in mind is that you do have continents moving, and that can influence the life forms that you see as well, the moving continents. And what happens here is this process is called plate tectonics. And tectonics is Greek in origin, which means to build. And so here you have your rigid slabs of rock on top. So you have both a lighter, less dense continental crust, and then oceanic crust, which is more dense and found in the lower areas where oceans settle in, or where the oceanic water settles there. And so as these continents are moving, the plates, they're separate plates. They move, they create earthquakes, they create volcanoes, even mountains when it's two continental crusts coming together over time. And so something to think about here is that these moving tectonics can also influence the evolution of life on the planet. Just having massive differences in where these continents are can create changes in climate itself. Rodinia is a supercontinent that there's evidence for. This Rodinia is, we see is going to form there at the end of the Proterozoic and then begins to break apart. Eventually coming to a Pangea here, and a Pangea, it begins to start to break apart maybe about 225 million years ago here. And as we progress, we get to our modern continents here. So from Pangea, we get directly to our modern continents, and the continents are still moving today. North America is still moving towards Asia on the order of centimeters per year. So the Atlantic Ocean is actually getting bigger, and the Pacific Ocean is getting smaller as time progresses. And so the connection now to, or correlating to what we see with life forms, not necessarily explaining for now, but noticing some of those big items here. First of all, life, you should know it emerged. Life emerged in the Archean eon. And so I want to make sure that you know that, again, that's 350 billion years ago. Do not confuse that with the formation of Earth, which is 4.6 billion years ago. And the other one that I want you to know is the Cambrian, where you have your Cambrian explosion. And so it's in the Archean where we see our first life forms. Do not get confused. Those early fossils that we have on our record to our formation of Earth. And then in the Proterozoic is the early life there. And one of the things that you're going to see is you already have your first cells forming, but one thing you start to see here is eukaryotes, which are more complex. And then multicellular organisms are also being seen here towards the end of the Proterozoic. Okay? And then the formation of Rodinia occurs during that time and then begins to break apart right after that. So when we get to the end of the Proterozoic, we're in the Phanerozoic. Know the Cambrian period as the first period there, and that's about approximately 500, half a billion years ago or so. You want to know that one because we do see the multicellular organisms evolving in those oceans, but it's during the Cambrian that we get what's called the Cambrian, or referred to as the Cambrian explosion. And here, it's called the Cambrian explosion because we have this massive diversification of multicellular organisms. And a lot of the multicellular organisms we see today can be traced back to the evolution that was occurring there. Now we progress further in time. We pass the dinosaurs here as they evolved there during the Mesozoic era. But we also see here that birds are actually descended from a line of dinosaurs. They're closely related to dinosaurs and crocodiles. So about 125 million years ago, you start to see some of the first flying birds, and that's only about 4% of Earth's entire time. And then we got to go all the way up here to see the appearance of humans, and that's only about 0.2% of Earth's history. So in this section, we're going to explore the origins of life. And one of the first things we're going to do is, as an objective, is to explore and contrast the difference between what is thought to have been the early atmosphere on Earth, which is considerably different than today's atmosphere. And then we want to make sure that we can kind of outline the key steps that would be necessary for the origin of life on this planet. Now, what we do know is that at some point we had to go from abiotic or non-living to biotic or living, and we know that the basic unit of life is the cell. So at some point cells formed. How that happened, we don't really know because it is not possible to generate an actual living cell in the laboratory. However, there are some clues, both observed in rock, the record of the rock, and as well as the through experimentation, as we'll see here in this section. So first of all, the idea, where did the early basis of life, the organic molecules, first come from? And that origin is not fully known, but there are some evidences that suggest that the origin could have been both extraterrestrial, from outside of the Earth, through meteorites coming down on Earth, and through terrestrial, or through formation here on Earth. So as it turns out, for the evidence for extraterrestrial origin, we do see that the Earth is receiving meteorites all the time, and comets as well may slam into the Earth early on, and they still do today. In fact, there was a meteorite that hit the Earth back in 2000, and when they analyzed it, they found many different types of organic compounds. I think somewhere upwards of perhaps 15% of the mass of the meteorite was organic compounds, so a significant portion of it. And this meteorite contained organic compounds like carboxylic acid, sulfonic acid, and even the amino acid glycine, which is used to make our proteins. Now, when they analyzed these carbon compounds, one question is, did the comet, when it hit the Earth, pick up the organic compounds with a tremendous heat involved from the surrounding? But when they analyze the carbon within these compounds, they find that the carbon isotopes, the isotope ratios, are not consistent. You might recall from Biology 1 that there are different forms of the same element. There's so-called isotopes. For example, there's carbon-12, which is the most common form, carbon-13, and the radioactive carbon-14. This would have the greatest proportion, but there is a certain proportion of those within naturally occurring carbon here on Earth. It turns out when meteorites, they look at the composition of meteorites, the carbon ratios are different, so that suggests that these carbon compounds form outside of the planet and not here on Earth. So the other possibility is that the organic molecules originated here on Earth. And how might that have occurred? Well, one thing is to consider the idea of what the early atmosphere was like, and one of the most popular views within science is that the early atmosphere was a reducing atmosphere. And the reason it was reducing is because it is thought that there was no oxygen. That would be oxidizing atmosphere. And hydrogen, a lot of hydrogen gas would have had to have been available. And hydrogen is a good donor, as we saw in Biology 1, of electrons. And anytime hydrogen itself, hydrogen gas, reacts with things, it gives its electrons to the other molecule. And whenever you transfer electrons to another molecule, that's known as a reduction. The opposite would be taking electrons away, right? So I just thought that the early atmosphere had carbon dioxide, nitrogen gas, water vapor, and some other types of compounds in the atmosphere as well. For example, methane gas might have been in there. And again, a reducing atmosphere because of the hydrogen gas ability to provide electrons in reaction. So one of the our group, a pair of investigators, Stanley Miller and Miller-Urey, did the experiment back in the 1950s, and there they assembled an atmosphere similar to the reducing atmosphere that we mentioned earlier. This atmosphere was placed above water, and the water was heated to not quite to boiling, but to just hot enough. And an atmosphere was cycled within this chamber, and then an energy source in the form of sparks to simulate lightning was done over a week's period. And this would be what the setup would look like. Here you see this round flask down here and this connection of tubing here. And so your heat source is down here. Here's your water to simulate an ocean. And as you heat things, when you get your convection currents, hot air, gases will rise and cycle through here. And again, within this closed chamber, you have that reducing type of atmosphere. You have those compounds that include nitrogen, carbon dioxide, methane, even water vapor cycling through there. And then you have your spark here to simulate lightning. And we're going to cycle this for weeks. And what you start to see is a buildup of these organic compounds, including even simple amino acids start to build up there. So you see from inorganic compounds that are here, you end up with these organic molecules. So this experiment provides evidence to support the idea that organic compounds can be formed here on Earth. So what they found on there? They found first went through a series of simple organic compounds, and then forming more complex organic compounds, including amino acids. Some further experimentation later on with similar setups produced over 30 different carbon compounds, including several amino acids, and even adenine. And we know adenine is found in adenosine phosphate, which is a component of DNA. So this is the base adenine. So again, we can see here that these key molecules can form here on Earth. So this brings up to the list, what are things that we would need besides just your basic organic molecules? You're going to need those to be present. Now, in order to have life, you have to have metabolism, and then you have to have a boundary, a membrane. Basically, you have a cell, a living cell. So we got to think, what was the progression of events that could have occurred? So if we just consider a metabolism, a basic metabolism, where compounds are made or broken down as energy is being used by the cell to do biological work, right? So there's two basic scenarios. One is an autotrophic origin, so making your own food arose first, and then a heterotrophic origin. So if we look at the autotrophic origin here, we got to think perhaps not necessarily photosynthesis, but a chemoautotrophic one, where were able to break down inorganic molecules, oxidize those, and then use them to drive a reverse of the citric acid cycle. The citric acid cycle, you recall from your biology 1 course, occurs in the mitochondria. As we oxidize food to produce ATP, we would be basically reversing that using the energy from oxidizing inorganic molecules. We do see organisms alive today that actually do that kind of stuff. Simple organisms like bacteria can do that, chemoautotrophic type of metabolism. The other origin may have been heterotrophic, in which you have organisms that are capable of breaking down organic compounds that could have just already been in the environment, whether it is through extraterrestrial or through the origins here on this planet. So there was plenty of carbon compounds rich in energy that could have been oxidized in a heterotrophic type. Which one may have occurred first is not known, but it makes sense that perhaps both were evolving at similar times during this time, because there's plenty of room for these types of metabolisms to evolve. But with that in mind, what are some landmarks in the evolution of metabolism? First was oxygenic photosynthesis. There's anoxygenic photosynthesis, where oxygen is not produced, but light energy is still used to produce. And it seems that there is evidence from the geological record, when you look at minerals and rocks, you can tell a point when there was free oxygen in the atmosphere and when there wasn't. For example, iron minerals. If you start to see iron oxide, which you might know as rust, in these minerals, then you know there had to be oxygen present in the atmosphere. And so there seems to be 2 main points in time, late in the Archean, which we discussed in the prior section, and another one late in the Proterozoic, where we seem to see these pulses of oxygen increase in the atmosphere. And the only real explanation for so much oxygen would be lots of microbes, like photosynthetic bacteria producing lots and lots of oxygen and releasing it into the atmosphere. Another one would have been carbon fixation, and that's where you take carbon, like as in the form of carbon dioxide, and then you bring it in and you make it a larger molecule and you reduce it to produce things like carbohydrates, which are excellent sources of energy. Another important landmark would have been the evolution of nitrogen fixation. Nitrogen in the atmosphere is N2, and it's a triple bond when you draw the molecule. That triple bond is very strong and very hard to break. And it turns out that the only organisms that are capable of breaking this to produce things like ammonia or nitrates, which are things that organisms can actually bring in and use to make things like DNA and proteins, which have nitrogen in them. They can't use N2 or atmospheric nitrogen, this molecule here. So you had to have organisms that evolve an enzyme. And today we know that there are bacteria that produce an enzyme called nitrogenase. And that one, there's some evidence to support that this particular enzyme may have evolved as early back as the late Archean, some 2.7 to 2.5 billion years ago. And why would this be necessary is because fixed nitrogen, getting it to go from atmospheric nitrogen N2, which is very stable, to these forms here, can be done by things like lightning or come out of volcanoes, but it just would not have been enough fixed nitrogen to support populations of organisms. So you needed to have the evolution of an enzyme that drives a metabolism that would produce these things. Now, if we build from these metabolisms, we're going to need a source of genetic a way of storing genetic information. That means information that allows you to replicate things in a regular way. And the question is whether it was DNA or RNA to first do this. The simpler scenario seems to be RNA for various reasons. And one of them is that RNA can actually serve the function as an enzyme, or has an enzymatic function. They call them ribozymes. This was something mentioned in an earlier chapter, perhaps. It was discussed during your 1406 course. And that means that the RNA itself can actually drive chemical reactions, reducing the activation energy, which is what enzymatic functions do. We know that proteins now do that today, but early on, perhaps proteins weren't around, and we had a molecule, RNA, that is capable of storing genetic information, because we see that today. It basically takes it can store genetic information and use that to reproduce things and even produce proteins. And start out with that one, and then from there evolve DNA, since it's more complex. And DNA is actually more stable. So the idea here is, why would this be a more favorable scenario? Is because RNA can also store energy and can serve as enzymes. Now later on, DNA becomes the choice, so to speak, for storing genetic information, and enzymes start to take over. Actual proteins start to take over the function of catalyzing reactions that occur within metabolisms. And some other evidences help provide more weight for this scenario. For example, RNA can spontaneously form given certain circumstances. For example, given the right compounds, organic compounds, on the surfaces of clay particles, which is discussed in your book. But the point is that RNA can spontaneously form and do these types of things that would be necessary for storing genetic information. The next thing you would need to do is be able to put all of this inside a controlled environment, and that means forming a membrane. And so the first life forms had to have been simple single-celled organisms. And what were the first membranes? They were probably made of fatty acids, which are amphipathic, which means they have both polar and nonpolar portions, and then eventually evolving phospholipids, which we see in modern membranes today. The point is that these fatty acids, because they have both polar end and nonpolar end, can form these bubbles within which you could corral these different kinds of molecules in there, where you're now housing a metabolism within there. So these molecules are now concentrated within a certain area, which means these reactions are more likely to occur. So the first organisms then that we see, our first life forms, had to have been these single-celled organisms. So in this section, we're going to look for evidence for early life, and the learning objective for this one is to evaluate the strength of fossil evidence dating the origins of life. So when we look at early Earth, the conditions under there, it seems likely that life on Earth evolved at relatively high temperatures. Your body temperature is roughly around 36 degrees Celsius, but the oceans 3.8 billion years ago were probably between 49 and 88 degrees Celsius. So that's quite a bit warmer. Remember, water boils at 100 degrees Celsius. So this would be scalding hot for us. So here, the first organisms probably evolved around this time, 3.5, 3.8 billion years ago. So we'll look at the evidences for that. Fossil evidence does indicate that life may have originated 3.2 billion years ago, maybe earlier. Depends on which evidences you look at. This would have been during the Archean, after the Hadean. And here we have to look at structures called microfossils. And these microfossils would be pretty small, about a micrometer in size. And to do that, we have to look at pretty ancient rocks that have been relatively undisturbed by the rock cycle. And such types of rocks are found in geological formations called cratons or cratons. And there's two that date back. The rocks in these cratons date back between 3.5 and 3.8 billion years ago. One is the Capival Craton in Africa, and the other one is the Pilbara Craton in Australia. And the structures in there that they found seem to be interpreted or can be interpreted as microfossils, or very simple cell-like structures that would be similar to, let's say, the prokaryotes we see today. Others include mat-like structures or mats of this biological film that might be left behind by certain types of prokaryotes, for example, cyanobacteria, photosynthetic bacteria. And this is one. This itself is not an individual cell. Just look at the scale here. The scale would be 100 micrometers. You could see this structure with your naked eye. It's about 1/10 of a millimeter in size. But in here, the structure overall is consistent with life forms that produce this film. And then when they look at this microfossil structure with transmission electron microscope, which sends electrons through there, you can see these little compartmentalized structures that again are consistent with simple life forms like the prokaryotes we see today. So these types of mat-like microfossils do date back about 3.5 billion years ago. Some other fossils that date back to 3.2 billion years ago do closely resemble larger eukaryotic cells. However, it's more likely that these were early cyanobacteria. And cyanobacteria are the ones that we see today that are capable of doing photosynthesis. They're prokaryotes as well. So we still haven't quite got evidence for eukaryotic cell types. Here's some indirect evidence called stromatolites. And these date back 2.7 billion years. And the thing about these is that these structures are consistent with more modern forms and more modern fossils that don't date back as far back as 2.7 billion years. It seems that cyanobacteria seem to deposit and keep some of the mineralized structures and deposit these formations that are called stromatolites. So these types of structures here are more consistent with being produced by life forms. So you don't directly see cells within these structures, or may not, but what you do see are structural formations that are consistent with what life forms do or have done on this planet. So again, modern forms of these that are not as old as 2.7 billion years have been found. So again, here are some more evidence of very ancient life forms called stromatolites. Now, another line of evidence comes from isotope data or isotopic data. And here, the idea is if you come across these microfossils and in there you find carbon compounds, and then you analyze their isotopes. And remember, isotopes have naturally occurring carbon-12. There's different ways to represent it. We can represent it with a dash in the mass number. Carbon-12, carbon-13, and carbon-14. And when you analyze these types of carbon and inorganic minerals found in rocks, you're going to see a certain ratio, certain proportion, a percentage of each one. But when you look at life forms, you see that when these life forms are fixing carbon, say they're taking in carbon dioxide and they want to produce organic compounds with it, they tend to favor carbon dioxide, in which the carbon dioxide has the carbon-12 isotope in it. So if you have a carbon dioxide that has carbon-13 isotope, it tends to get ignored by the metabolism and doesn't get fixed, right? So what does that do? That skews the ratio or makes the ratio different when it comes to organic compounds that are produced by living things, right? So what you do is you look at these microfossils and analyze the carbon compounds within there, and you find that the ratios are more consistent with carbon fixation from ancient life forms. And when they do that, they found these types of compounds in rocks that date back 3.8 billion years ago. So here the organic compounds there suggest that carbon fixation may have started back as early as 3.8 billion years ago. Now this is before those ancient fossils that we were looking at a while ago, where those date back 3.5 billion years ago. So for now we set the most ancient life forms around 3.5 billion years ago overall. So when we look at what are ways in which carbon may have been fixed, we look at clues from life forms we see today, and it seems that today's modern bacteria, all of them are that do run they run a metabolism called the Calvin cycle, which we studied in photosynthesis in your 1406 course or your biology 1 course. This is the one that occurs within the stroma of chloroplast, but it seems that when it comes to cyanobacteria, green plants living today, and even bacteria that do use photosynthesis, but don't produce oxygen, anoxygenic, like purple sulfur bacteria. They all use the Calvin cycle to fix carbon dioxide. And then we also see some archaea that use a reverse form of the Krebs cycle to fix. Krebs cycle actually produces carbon dioxide as it the normal Krebs cycle, which occurs in the mitochondria. The Krebs cycle is also the citric acid cycle, but if you reverse it, you would be bringing carbon dioxide in. So that would be a reductive version here. And we do see lithotrophic or bacteria that basically break down rock or oxidize rock to drive this. This was mentioned in the prior section as well here. So basically, these are two ways we can fix carbon to make organic molecules. That's what life does. And when these metabolisms are doing this, they tend to favor the carbon-12 isotope. So that's where we say we're going to see ratios that are different when these organic molecules are of life origin. They're produced by living things. Okay. What this also tells us is the ability to fix carbon has evolved more than once. So in other words, and we see that with the different metabolisms that are capable of fixing that carbon. We also might look for other indirect forms of life by looking for compounds that seem to only be produced by life forms. For example, hydrocarbons. When you find hydrocarbons, these are going to be things like fatty acids, for example, that they have these long chains of carbon on there that have hydrogen in there. And so when you study organic chemistry, you see, well, the hydrocarbons are these compounds that just have these long chains. Sometimes they're ring-like structures, and everywhere there's a carbon, there's hydrogen attached to them. That shows that carbon is reduced because you have your hydrogens with their electrons on there. And so when we look for evidence of that in rocks, right, that would be an indirect evidence because these are compounds that are typically there's no other explanation for their origin other than life forms producing them, right? So the evidence for this is difficult to find, but they have found hydrocarbons like those fatty acids we were talking about there, and they're consistent with cyanobacteria that dates back 2.7 billion years. And when they analyze these hydrocarbons, they also seem to have those carbon isotope ratios that also indicate fixation by living things, so that biological carbon fixation, again providing more evidence for ancient life forms. So this section looks at Earth-changing systems, and in this section, the learning outcomes include being able to construct an explanation for the relationship between CO2 levels in the atmosphere and glaciation, or the formation of sheets of ice on the surface of the Earth, and to be able to argue how plate tectonics can affect the evolution of life on Earth. So one of our systems includes climate. Climate is long-term trends in both temperature and water availability, and that's connected to the atmosphere, especially levels of CO2. Carbon dioxide, we talked about in prior section, is a greenhouse gas and can trap heat. So you have increase in CO2 levels, you have a warming that would occur of the planet. If you have a drop in CO2 levels, then that would be a cooling effect. And when you have dramatic shifts in these factors, climate and the relationship with atmospheric levels of CO2, when you have major shifts in climate, that can lead to mass extinctions. And mass extinctions do change the course of evolution on the planet. Now we know Earth's climate has been changing since the beginning. But one thing is true is that when you have extreme drops in temperature, that's going to result in glaciation or glacial ice, and that glaciation can lead to mass extinctions. So there's something to think about there overall. Now, this diagram here does show the entire history of the Earth going back 4.6 billion years ago. And the scale here is in the millions of years, so 1,000 million years is a billion years. And we're focusing here on the Proterozoic time. And so on this graph, Proterozoic would be the eon defined by that period of time there, from about 2.5 billion years to nearly 500 million years ago. And you can see that the dark blue lines represent periods of time in which we have massive glaciation from pole to pole called snowball. Earth is the nickname for that. And then when you have less periods where you have significant cooling, but you don't have glaciation from pole to pole, but regional glaciation. And sometimes we may refer to these as ice ages, for example. You can see the time frames there. And so when we look at the geological changes that occur and the correlation in changes in living organisms, when you have, say, for example, mass extinctions and so on, some of these can be explained by shifts in the Earth's atmosphere. And when you have shifts in Earth's atmospheric CO2, you're going to have changes in the level of warming or cooling that occurs. And so during periods of snowball Earth, later in the Proterozoic, which would be these two snowball events here, the continents were in the tropics. And when you're in the tropics, you're receiving more direct sunlight there. And so you imagine this is a period of time when this is before snowball, so you're leading towards a snowball event. Things are warm at that time. When it's warm, you're going to have wetter climates. You're going to have more atmosphere, more water in the atmosphere. That means more rain. You have more rain, and that rain combined with CO2, we talked about in the prior section, the CO2 reacts with water to produce carbonic acid, which then weathers the rock, and then that weathered rock releases minerals that contain calcium, and then calcium reacts with the bicarbonate, as discussed in the prior section, and combines with it to form calcium carbonate, which precipitates to form things like limestone rock, sedimentary rock. So basically that captures CO2 from the atmosphere, lowering it, and what that results is in cooler global cooling, and that results in glaciation. So once the glaciation appears and cooling occurs, you're going to have a decrease in weathering, and that has an effect of stabilizing temperature overall. And then you'll have another warming period because for some other geological event, perhaps a volcano releases more CO2 into the atmosphere, creating a situation where you have more warming, and then the glaciers retreat or go away or melt away. So the plate tectonics themselves can also influence how much weathering occurs, and this is because if you have one larger continent, then you have a certain amount of shoreline. But if the continents begin to break apart, you're going to have more shoreline. You're going to have greater amount of shoreline, which can create more opportunity for more weathering to occur, and that can have an accelerating effect on weathering, which can accelerate cooling because the weathering leads to CO2 being captured and converted into calcium carbonate. So the continents, the motion of those continents can be explained by plate tectonics, which was mentioned in the earlier section there. And those shifting plates themselves can influence evolution, because when you see this trend where you go from a larger continent right here, like Rodinia, and then the continents separate, and then they come back together again, like you see in Pangea here, and then they separate again. You're basically going to be separating populations of terrestrial organisms, whether it's plant life, animal life, and then they get separated, and so that's going to allow evolution to occur in on different land masses. And then the land masses come together, and now you have these organisms moving back and forth, and then they start interacting with each other, and then that can create more evolution, and then they separate again, and so on. So that's the moving continents themselves can have an influence on evolution on the planet. Now, so you can track those events there. They do mention specifically during the 700 million years ago that supercontinent Rodinia begins to break apart. And then eventually we get to Pangea here. Pangea forms there at the end of the Paleozoic, and then about 225 million years or so, you start to see that Pangea begins to break apart again, forming some of the continents we see today. The point is that coming together and moving apart can bring populations together or separate them, and that influences evolution. During the Cambrian, that's one of the times or periods I want you to know. The Cambrian is early in the Paleozoic. I cut off the bottom part of the graph here, so we don't see the Hadean or Archean in there. But during the Cambrian, you have that Cambrian explosion, which was mentioned earlier. There you're going to have a great amount of evolution occurring and diversification of multicellular organisms. And then you also have the corresponding separation of the continents during that time. The Paleozoic is going to end here. So the Paleozoic is one of the eras there. It's going to end right here with the formation of Pangea, mentioned already. And then we start to see the separation of those continents overall. When we get to the Cenozoic, which is the modern era of life, you start to see the familiar land masses that we see today. Australia, Antarctica separated, Greenland separates from North America. And so again, all of these movements again can influence evolution. Now during the Cretaceous here, the Cretaceous is a period within the Mesozoic here. At the end of the Cretaceous, we get the extinction of the dinosaurs, and the evidence strongly suggests it was the impact of a large asteroid. But at the end of the Cretaceous there was a significant rise in greenhouse gases, and so there was a warming period during that time. And when you have that warming, you start to see more of the glaciers retreating. And you even see some of the polar ice, the ice at the north and the south poles begin to melt more, and that creates sea level rises to the fact that you're to the point where you get inland seas. In fact, there was an inland sea even coming through Texas here. So when you go and you look at parts of central Texas, there's fossils of sea creatures in those rocks there that once lived in the oceans that existed on there. So that's pretty interesting. And so you can imagine when sea levels rise and then they retreat again, and you no longer have oceans, you're going to have a whole bunch of populations dying out. So populations, perhaps even whole species, going extinct. And in the last 2 million years, there was what we may have referred to as an ice age as well. So you have another regional glacial period. A lot of North America, the northern hemisphere, was covered in ice. And that caused sea levels to drop to the point where you have the formation of land masses. That's pretty important because that allowed animals and plants to move. Animals are good at moving plants around, where they're carrying seeds in their gut and then release them elsewhere. So you have the movement. You have a movement here. This land bridge connection here between Asia and North America. There, the Bering Strait. And this includes humans too, because humans had already evolved by then, and so they're moving. And we also have another land bridge forming between Asia and Australia. So you have all of this movement of animals and even plants, and that influences the course of evolution as well. So life itself can influence evolution. So this interplay between geological and living things influence the course of evolution. And one of those major changes that occurred when it comes to life, the evolution of life, was the oxygenic photosynthesis, where photosynthesizers are actually splitting water during the process to release oxygen. There's anoxygenic photosynthesis. Anoxygenic means without oxygen. And here we're not releasing oxygen. But the photosynthesis you likely studied in some detail in your Biology 1 or 1406 course showed how your photosystem 2 splits water to get to replace the electrons that were lost from the photosystem there. And so when you have this oxygenic photosynthesis, you're going to start to have a buildup of oxygen in the atmosphere. However, there was a 200 million year lag time during one of the first surges of oxygen there late in the Proterozoic, and that would be about right there. And so there's going to be so the scale is in the billions of years here. So you have this maybe 200 million year lag time, and that's because some of the first oxygen being released, likely by cyanobacteria and maybe some algae, releasing oxygen. Well, that oxygen wasn't just free to go anywhere because there was a lot of iron in the minerals that were present at the time. And without oxygen, the iron does just fine. But we know that if you have just iron out in our atmosphere right now, it starts to rust just spontaneously. It'll begin to react with the oxygen. So as that oxygen was releasing, it was combining with the free iron that was available, and it took probably 200 million years to use up most of that iron. And then you start to see a significant increase right about this point in the atmosphere. And then there was another surge about at this point here. And this is significant because having more oxygen in the atmosphere allows for us to have be able to use oxygen in aerobic respiration. So that allows for more of that kind of metabolism to occur. And also having oxygen in the atmosphere leads to the formation of ozone. As the oxygen makes it up to the stratosphere from the troposphere where it was created, we have O2 going up there, and molecules of O2 are going to react with ultraviolet, and that breaks the bonds between the oxygen atoms, and then new bonds are formed to form a molecule with 3 oxygens, and that's your ozone there. And so this reaction goes back and forth up in the atmosphere, and that reaction is actually what helps us. Let me balance this here. That helps us because the ozone up there and the formation and breaking, the making and breaking apart of ozone actually helps absorb 95% of the ultraviolet, which would otherwise damage sensitive molecules like DNA, making life on the surface of this planet impossible. Ultraviolet is very damaging, and you know that already. If only 5% can give you a significant sunburn. And so when it comes to how light can influence changes that can occur, right? The interplay between non-living and living aspects of Earth systems here. There's a growing body of evidence that strongly suggests that plants also contributed to the glaciations themselves. So if we look at both plants and animals, their origins are in the water, out in the ocean. So when we look at plants colonizing land, the first wave of plants to colonize land were simple plants. And this occurs during the Ordovician period here. So the Ordovician is about right here. And you have some simple plants that are first moving onto land. As they move onto land, they begin to diversify, colonize, and become the dominant living things that are capable of photosynthesis. And what does photosynthesis do? Well, photosynthesis is going to take CO2 out of the air. We also need water. We're going to split water to generate oxygen and basically produce organic molecules like glucose, C6H12O6, and release oxygen. And so basically you're pulling CO2 out of the atmosphere, and we know when we lower our CO2 levels, that can create a cooling effect. So there was a cooling period that resulted in a regional glaciation during the Ordovician, and then vascular plants, which we're going to be studying later, are more advanced plants, evolve and then become dominant on in terrestrial environments. So we have the early vascular plants are beginning to evolve and diversify, and then they begin to dominate during the Devonian period here. And that second wave here, again, more photosynthesis, decrease in CO2, and we get another cooling period. Another thing that can happen is that these plants do release acids, organic acids, into the soil, and organic acids can help dissolve more rock minerals, and that causes more weathering. And we already know what weathering does. That weathering can also reduce the amount of CO2 from the atmosphere, because the CO2 basically ultimately reacts with water that produces carbonic acid, which then reacts with minerals that have calcium to form calcium carbonate, and then the CO2 now is removed from the atmosphere in that way. And so what ends up happening here is regional glaciations that causes drops in sea level, and that can create some extinctions, which influences the course of evolution. So your objectives in this section that discusses how life has changed on this planet and actually continues to go through changes or evolves. It includes being able to compare and contrast the evolution of the endomembrane system and mitochondria. How did mitochondria and membranes inside evolve? And then discuss the types of specialization necessary to get multicellularity. And number one is you got to be a eukaryote, right? So when we look at life that we see today and the history of life on the planet, it seems that life evolved into three monophyletic domains. They include the eubacteria, or the true bacteria, or common bacteria, and the archaea, the group we find extremophiles, and then the eukaryotes, which are in the domain eukarya, right? And the eukarya are displayed here in this diagram here in purple. These are all eukaryotic groups, and we're going to divide those into 6 supergroups. These supergroups are not the same as kingdoms, and the supergroups are more for us to kind of keep a better accounting for phylogeny, as there are issues with classification and taxonomy that we discussed in the prior chapter. A lot of times there's good matches in how we've classified organisms here, but the supergroups are created to help take care of the problems we see with our current classification system. There are 6 supergroups which are going to be covered as we progress through there. There's the excavata, and these have atypical mitochondria. The mitochondria have gone through some changes, loss of some functioning, and so on. And we'll see that when we look at the excavata. The chromalveolata have, if they do have chloroplasts and do photosynthesis, they were taken in by a process called secondary endosymbiosis. Talk about that in a little bit. The archaeplastida, these are your other photosynthetic organisms. They include algae and land plants, and they also, all of them have chloroplasts in this group. That's not true for the chromalveolata. Then you have the rhizaria, and what characterizes the rhizaria is slender pseudopodia. Pseudopodia translates to false feet. And then we have the amoebozoa, and the amoebozoa have blunt pseudopodia. And pseudopodia, when you look at these, are aquatic organisms, and pseudopodia are basically extensions of the cytoplasm, and the organism is capable of moving the cytoplasm around. It allows them to crawl on surfaces and to even swallow food particles through a process called endocytosis. And then you have the opisthokonta here, and the opisthokonta includes the fungi and even the animals, and the name itself translates to mean, opisto means rear or posterior, toward the back, towards the rear, and konta means an oar or a pole. And this is a reference to these organisms producing at least one cell type that has a flagella in the back, which means it pushes the organism through the water or through the medium. And so a key here, when we talk about a distinction between prokaryotes and eukaryotes, the key is going to be compartmentalization, being able to separate the metabolisms that are occurring within your cytoplasm. And bacteria, the key here is that they lack compartmentalization. They're relatively simpler in structure. That doesn't mean their metabolisms are not complicated. But the bacteria and the archaea don't have any real compartmentalization. They don't separate with membranes within their cells, roughly. And these guys were around for and dominating for about 1 billion years. Remember that the first prokaryote evidence occurs about 3.5 billion years. And then in order, we don't see eukaryotes appear in the fossil record until about 1.5 billion years ago. So for the most part, you have prokaryotes, and then the eukaryotes evolve later. And the key here for the eukaryotes is that they have this extensive endomembrane system, which includes the nucleus and the endoplasmic reticulum. And what this does is it allows us to compartmentalize your metabolic processes. And when you can do that, when you can separate your metabolism to different compartments, you're more complex, right? And what this allows, the compartmentalization allows larger cells. Typically, a eukaryote is going to be about 10 times bigger than a prokaryote. And this increased complexity leads directly to multicellularity, which means you have organisms made not just of more than one cell or many cells, but many different cell types. For example, your body has, the human animal has 250 different cell types. You're made of trillions of cells, but you're multicellular because you have at least 2 or more cell types. So what is the endomembrane system here? The endomembrane system is thought to have arisen from infoldings of your plasma membrane, right? So you imagine here you start off with a cell, you have your chromosome there in the middle, and then this plasma membrane begins to fold inwards. And we see bacteria that are like this today. They have a lot of infolding of the membrane, but they just don't quite have this level of complexity. And so that membrane begins to fold inward and then surround the chromosome and then completely seals it off. Now you have a nucleus, and then you have this folded membranes that become things like your endoplasmic reticulum and your Golgi apparatus. And so going back to the nucleus itself, the nucleus here in the center where your chromosomes are, remember your chromosomes have the instructions for running the show of the cell. They have instructions for building a cell and for producing enzymes that run metabolism in there. So the control is done by the DNA. Your DNA has the codes, and those codes have to be transcribed, a process called transcription. Okay? And they're going to be transcribed into RNA, the codes. And RNA, they're both nucleic acids. We're just going from one form of genetic storage to another. And then that RNA then goes and attaches with ribosomes to translate to make your proteins. Okay, so this process here, defined by that arrow, is called translation, right? Well, if you're a prokaryote, there's no boundary, there's no membrane that separates this, right? As soon as your RNA is produced, the ribosomes attach and begin translating into a protein. But if you're a eukaryote, and we see this electron micrograph here, your chromosomes are in here, your DNA is in here, so here transcription occurs here. You go from DNA to RNA, and then your RNA has to go outside of there. So already you see a compartmentalization that separates the transcription from the translation. That's a level of complexity you do not see in prokaryotes, right? And then when you have these other membranes here, the endoplasmic reticulum, which probably started off as enfoldings themselves, and your Golgi, this is going to help, these structures help not only produce things separate from the rest of the cytoplasm. In the rough endoplasmic reticulum, we make proteins, for example, that we don't want made freely in the cytosol or the cytoplasm. You produce them there, and then you transport them and modify them further. You put them in a transport vessel. They go to the Golgi apparatus and get modified further. And they may produce organelles called lysosomes that stay in there, therefore intracellular digestion. Or they may produce vesicles that go for secretion, and so the cell eliminates the materials outside of the cell. And so all of that makes for a more complex functioning within the cell. Overall. Now, that's your endomembrane system. So there are going to be other organelles in there that are not derived from this endomembrane system. And in fact, they're derived from other organisms that ended up being taken in by a process called endosymbiosis. And so the mitochondria and the chloroplasts, their origin is different. And there's lots of evidence in the mitochondria and chloroplasts we see today that support this. They have, for example, their own ribosomes, which are smaller than eukaryote ribosomes. These ribosomes are the size of prokaryotes. They have their own circular DNA, and you only see circular DNA in prokaryotes. These mitochondria and chloroplasts self-replicate. So there's several supporting lines of evidence that you see in mitochondria and chloroplasts alive today that are parts of living cells today that strongly support the endosymbiotic or endosymbiosis theory. The mitochondria are thought to have arisen from or descended from relatives that are related to purple surface bacteria. And indeed, today you see certain bacteria that can live inside of eukaryotic cells, for example, the parasite Rickettsia. can live inside of a eukaryotic cell. Now, it actually harms the cell that it lives in, but living endosymbiotically, a smaller cell living inside a larger cell is not unheard of. So you can imagine a situation where a cell takes in a aerobic bacterium, and then instead of digesting it down, starts to live together with it, and then over time the bacteria can no longer live on its own, and the two have developed a dependent relationship on each other. In that case, that would be primary endosymbiosis. Okay? Now, when it comes to chloroplasts, initially chloroplasts were probably a cyanobacterium. We see cyanobacteria today, they're photosynthetic. They release oxygen as they do their photosynthesis thing, and metabolism that's pretty much the same as we see in land plants today. So the bacteria gets taken in. You already have your mitochondria in there by some eukaryotic cell. Gets taken in, and then the cyanobacterium no longer lives on its own, and that gives rise to the archaeplastida, where you have your green algae and your red algae that we see alive today. And then you have brown algae, and the brown algae, there's a lot of evidence to suggest that the brown algae arose by a secondary endosymbiosis, where in the secondary endosymbiosis, you already have a eukaryote red algae ancestor. So there's red algae still alive today, but certain red algae may have been taken in by a non-photosynthetic eukaryote. So you're taking in another eukaryote, goes in there, and then instead of digesting down the eukaryote that was photosynthetic to red algae, the two begin to establish a relationship. And then over long periods of time, the eukaryote red algae starts to lose its nucleus and other components. And now you have a chloroplast that, instead of having two membranes like the original chloroplast, now it has three membranes or multiple membranes. And then that highlights or provides that evidence that supports this secondary endosymbiosis. And that's going to be characteristic of photosynthesizers that are in the supergroup called chromalveolata. So what are some components here or some things that are going to allow us to become more and more complex as eukaryotes? Well, first of all, we need to recognize how important being unicellular is. Being unicellular, like that amoeba we saw earlier, and all prokaryotes, your bacteria and your archaebacteria, they're all unicellular. Some of them live in colonies and maybe even communicate with each other, but they're not multicellular. In order to be multicellular, you have to have different cell types within that same organism. So you have to have those sorts of changes occur there. Now, unicellular are very successful. We still see them today. In fact, they make up most of the biomass on Earth, unicellular prokaryotes and eukaryotes, right? But there are limits onto how much these unicellular organisms can evolve. So what has to happen is you start living in a colony, and here you see a colonial organism. They give it a scientific name, but that's we don't need to distract ourselves with what the name is, other than this is a colony of eukaryotic cells, and they are communicating with each other, but every cell in that colony is doing the same thing. There's no specialization here. In order to get multicellular, not only do you have to be colonial, but you have to have cell differentiation occur. So at some point living in a colony, some cells begin to start doing different jobs. And every cell type that's in there is going to have the exact same genetic makeup, but they start differentiating. And in order for that differentiation to occur, you have to basically turn on different sets of genes. So every cell has the same set of genes, same sets of genes, but you turn on different sets of genes, and you're going to get different cell types. So once you start getting simple multicellular, then you start getting more and more complex. You start getting tissues, and then organs, organ systems, and then very, very complex organisms like you see in animals, and even plants can be quite complex. So one of the things we see here is, first of all, you're not going to see multicellular in prokaryotes, so no multicellular there. Multicellularity is seen within your eukaryotes, and not in every group, but in many of these groups. And one thing that is certain is that every one of these groups that we see here, their ancestors were unicellular. And what that tells us is that multicellularity evolved independently of multicellularity in other groups. In other words, multicellular plants evolved independent of multicellular animals. We didn't get multicellular, and then from there, that common ancestor gave rise to plants and then animals. Multicellular arose differently, independent in each group. That's true. And so, again, you're going to have within these groups them starting to live in colonies and communicating, and then from there, differentiation occurs. And that now within the body of a multicellular organism, say an animal like a human or oak tree, all of the cells within the body of that organism are genetically identical. Okay, but the key here is flipping on different sets of genes so the different genes are expressed. And so then that gives you different cell types and specialization. Now, what we also see in eukaryotes is the evolution of sexual reproduction. And sexual reproduction is important because that gives rise to genetic diversity. And genetic diversity is important when you have a planet in which climate and the environment is constantly changing. If you're genetically diverse, you can evolve over time more easily. If you have a population of all clones, then if the environment changes significantly and one individual can't handle it, then they all can't handle it. You're more likely to go extinct that way. Now, sexual reproduction does allow this genetic diversity, and in order for that to occur, a process called meiosis had to evolve. Now, in order for that to have happened, we first have to have the evolution of diploid cell types. It seems that if we think about it, the first eukaryotes, they were probably haploid, and that means they had only one set of chromosomes, right? At some point, diploid arose, and probably arose multiple times independently within different groups. And so you have diploids, and the diploid condition here is where you have two full sets of chromosomes. And if you go through normal mitosis is not going to give you genetic diversity. You're going to get two genetically identical cells with one cell division. But when you have meiosis is going to go through two cell divisions, and ultimately you're going to end up with, if you go from one cell, starting with one cell through two cell divisions during meiosis, you're going to end up getting four cells, and each of the four cells, they're all haploid now because you've reduced the number of chromosomes. But in that process of meiosis, includes events like crossing over that leads to genetic diversity. These were things that were covered in your Biology 1 course. Once you have your haploid cells, in animals, these would be gametes and sperm and egg cells, and they would unite to reform your diploid condition for your multicellular animal. The thing is that each one of these gametes is genetically different now because they're going to have different combinations from the prior generation. The generation before had a set of chromosomes, one from each parent. And so any one of these gametes, if their sperm can fertilize an egg, and since they're genetically different, then you're going to get offspring that are genetically diverse. This is just kind of a review of some of the things that occur. First of all, if you have two sets of chromosomes that are paternal and maternal, they can line up independent. This is for a simple model where you only have two pairs of chromosomes. Humans, for example, have 23 pairs, so not just two pairs, but 23 pairs. Just with two pairs alone, you're going to get 1, 2, 3, 4 different combinations just from the way the chromosomes line up, the pairs of chromosomes. That's called independent assortment. One pair lines up independent of another. When it comes to humans, we're talking about two possible ways of lining up for 23 pairs. So that's 2 raised to the 23rd power. That's over that's going to be over 8 million. So you're going to have approximately 8 million or just over 8 million combinations just with independent assortment. And then for every pair, you end up getting crossing over. So here, when the pairs line up during meiosis 1 on the metaphase plate, even before they even line up during prophase 1, you're going to get crossover events where you're going to have pieces of maternal and paternal chromosome exchange with each other between non-sister chromatids. And so you end up with gametes where you have chromosomes that have pieces of paternal and maternal, and that increases genetic diversity even more with the gametes that are produced. And then at the end of the day, you have genetically different sperm and eggs, and then if fertilization is random, that increases genetic diversity in the next generation even further. So just a quick review of some of those concepts. Now, during the evolution of multicellular organisms, that occurred before the Cambrian explosion. The Cambrian explosion is, or the Cambrian period occurs right at the beginning of the Phanerozoic, and just before that, in the Proterozoic, you have simple multicellular organisms already have evolved, and they evolved in the oceans, right? And then during the Cambrian explosion, you have this massive evolution of multicellular organisms, mostly aquatic, during that time. So during that 50 million years of that Cambrian explosion, in the animal group, for example, most of the ancestors that gave rise to the animal groups we see today appeared during that time. And so if we look back before that Cambrian explosion, there was 3 billion years where life was mostly unicellular going before the Cambrian explosion. Remember, for about 1 billion years, there's no evidence of any life, and then you start to see it about 3.5 billion years the first prokaryotes evolve. So that's about 3 billion years before the Cambrian explosion occurred, mostly unicellular. And so you have multicellular eukaryotes. You got to be a eukaryote to be multicellular. There's no multicellular prokaryotes. So if your origins are in the water, you do have to move on to land. And some events that occurred prior to that were pretty important before colonizing and moving on to land. So after the Cambrian radiation, you got to move on to land, but one major event occurred before that. Now, the use of radiation just means the evolution and diversification of groups of organisms. For example, when we talk about later on when the dinosaurs go extinct, there were already mammals present, but the dinosaurs were dominating the Earth, both terrestrial and aquatic. And when they go extinct, that allowed mammals to go through their radiation. They went through evolution and diversification. So radiation that's how it's used. So during the Cambrian explosion, you have a radiation. You have this massive diversification. But some key events occurred before then. We talked about this earlier. The evolution of oxygenic photosynthesis is what led to that ozone layer. Remember that ozone is responsible for protecting life on the surface from damaging ultraviolet. The ozone blocks 95% of ultraviolet radiation. If it wasn't for that, life on the surface would not be possible. It would need to have remained in the water, where the water does protect from ultraviolet radiation. Now, when you move from water onto land, you have to be worried about drying up. So you want to prevent desiccation or drying up. And in animals, vertebrate animals anyway, the amniotic egg evolved. The amniotic membrane prevents the embryo from drying up. Before that, fish, amphibians even don't have this, and they have to have their eggs laid in the water and fertilize in the water and develop in the water. For plants, the evolution of the seed did the same thing. Another way of solving the problem of protecting the embryo is the evolution of the seed. We have to obtain water, or have ways of obtaining water. In the animals, vertebrate animals, the evolution of the kidney is a way to help us, as we take water in, help us prevent too much water loss as you try to get rid of your metabolic waste products, which can build up in your tissues and become toxic. And then we also need a way to exchange gas with the atmosphere. In vertebrate animals, for example, We have lungs and other kinds of animals, other means of exchanging gas. For plants, land plants, you have the stomata within the leaves, the little holes that are under there that allow them to exchange carbon dioxide and oxygen for purposes of photosynthesis. So these are ways that your multicellular organisms solve problems of moving onto land. So be familiar with those.